Assalamualaikum and greetings everyone. Welcome to our journey into the fascinating world of the enzyme-linked immunosorbent assay, or known as ELISA. In this video, we'll break down the ELISA process step by step, explore its different types and see how it's used in real-world applications. What is ELISA? It is a powerful and widely used technique to detect antibodies, antigens, proteins and hormones. These are the four main types of ELISA. Firstly, we have direct ELISA, in which this method involves an antigen being adsorbed onto a microplate. An enzyme-labeled antibody specific to the antigen is then added. Next is indirect ELISA. Here, the antigen is also adsorbed onto a microplate, but instead of an enzyme-labeled antibody, a primary antibody specific to the antigen is added first. Then, a secondary antibody that is enzyme-labeled and specific to the primary antibody is introduced. Now, let's look at sandwich ELISA. This type requires a capture antibody that is adsorbed onto the microplate first. The antigen binds to this capture antibody, and then a second enzyme-labeled detection antibody binds to the antigen. Sandwich ELISA is highly specific and sensitive, making it ideal for detecting antigens present in low concentrations. Finally, we have competitive ELISA. In this method, the sample antigen competes with a labeled antigens for binding to a limited number of antibody sites. Now that we've explored the types of ELISA, let's delve into why ELISA is so significant and relevant in today's world. In clinical diagnostic, ELISA is used to detect infections like HIV, hepatitis, and COVID-19, as well as conditions like allergies and autoimmune disorders. It's also used in the food industry to ensure safety and quality by detecting allergens and contaminants. So, what makes ELISA such a preferred method? Let's look at some of its key advantages. First, ELISA is highly sensitive and specific. It can detect even minute amounts of antigens or antibodies in a sample. Second, it's versatile with different formats like direct, indirect, sandwich, and competitive ELISA to fulfill various needs. Third, it's relatively quick and easy to perform. Additionally, ELISA is cost-effective and it can process many samples simultaneously. However, like any technique, ELISA has its limitations. One major limitation is the risk of false positives or false negatives, which can occur due to cross-reactivity. ELISA also requires careful handling and can be time-consuming, and also it requires well-defined antibodies, which might not always be available. In summary, ELISA remains a powerful technique in diagnostics and research, which makes it a go-to method for many applications.